Randi Vita. I'm going to talk about how you can use the query interface, and I'm going to walk through the reporting interface using a user example. So this is the IEDB homepage, which we hope you'll be very familiar with after the end of this workshop. It has these 6 key search panes, and I'm going to walk through each one in detail. And they have 7 main search components. And this is where 99% of the queries done on the IEDB can be performed directly from this homepage. So we tried to put everything that people use the most often right here on the homepage. So starting with the first box, epitope, the details of this search box are that you can use the radio buttons to limit to linear, discontinuous, or non-peptidic epitopes. Or if you have a peptide of interest, you can put that peptide directly in to this text box. But I wanted to point out that another author who may be studying what is essentially the same epitope as you, they may have a slightly longer or a slightly shorter peptide, or they may have altered 1 or 2 of the amino acids, or maybe a slightly different variant. So I suggest always starting with a more broad search and using one of these blasts, 70, 80, or 90%, to include a larger amount of data. And then if you have a ton of data, you can narrow it after the fact. But this opens you up to what other authors might be thinking. My button stopped working. And then the next search pane is about the epitope source. So if the epitope is natural, it will come from an antigen, and that antigen will come from an organism. We do have data where the epitopes are not natural, so they won't have any source, so you wouldn't use this search box to access them. So if you want an organism, you can just start typing, and it will autocomplete for all of the organisms that match what you've typed, for which we currently have data, and this changes over time. So we may not have had a lot of coronavirus data prior to the pandemic, but now we do. And then the next part of the source is the antigen. So this is typically the protein, but it can be a larger non-peptidic structure that the epitope comes from, and you just start typing, and it'll again autocomplete with all of the other proteins that match what you typed. But a word of warning that many different species have proteins of the same name, so it's a lot to wade through. So many viruses will have a capsid protein. So if you just start with the organism search, you can then later very quickly and easily filter to just the proteins from that organism, and I'll show that when I get to the results page. So the next search box is the host. This is whose immune response is being studied, whose TCRs or antibodies or MHC molecules were in the experiment. And the radio buttons give you some key human, mouse, or non-human primate. But just like the other search boxes, you can just start typing, and you can use common names like dog or human, or you can use scientific names. And for all of the search boxes, there was this find button. And when you click on the find, it opens the finder relevant to that type of search. So this is the host finder, and we use those external ontologies, and that gives you here, in this case, the NCBI taxonomy hierarchical tree. So you can browse through and see all the different types of vertebrates for which we have data, and that also changes over time as new species get published having adaptive immune responses. They'll show up here, and you may be surprised at all the different species we have. And then the next search box up at the top is the assay. So we have T cell, B cell, and MHC ligand assays. And if you know a specific assay type you're interested in, you can start typing it, and it'll autocomplete. But it's often better when you first come to the IEDB to browse the tree and see what types of assays we have data for so far. And when new assay types come out in the literature and get developed, we will eventually get them added to the tree. But if you have an assay type that's only appeared in 1 or 2 papers, and we may not add that exact precise assay type, so you can go with a larger grouping, like a biological activity assay, 3D structure assay, binding constant assay. You can search on a high level or a very low level. Look and see what we have. And then the next search pane is MHC restriction, and this is applicable to T cell assays and MHC assays. And for you can use the radio buttons to narrow to just class or non-classical, or you can start typing. And because MHC molecules, the nomenclature is really complicated with dashes and asterisks and colons, it might be better to browse the tree and see all the different MHC molecules for which we have data. And just like the others, this does change over time. We will gain new species as they come up in the literature. And you can also here, just like the other trees, search on a high level or go very low. So you can do all human class 1 or you can do AO201, depending on what you want to see in your results. And then this is the last search pane on the home page. It's for the disease. And this is the disease state of the host at the time of the experiment or prior to the experiment, if they were cured or recovered, if it's relevant to the adaptive immune response being measured in the assay. So if someone had a malaria infection and you're measuring Plasmodium falciparum epitopes, that infection is definitely relevant to the immune response. And just like the others, the find takes you to a ontology tree. This is the disease ontology, and you can open it and navigate through and see all the different types of diseases we have. There was a question earlier whether we had autoimmune diseases. Yes, we do. Any type of disease that comes up in the literature that's relevant to the adaptive immune response will be captured. And just like the others, if a new disease comes up that there's published data for, it'll show up in the tree with the following build. So next I'm going to do a query from a user and show you how to use the search interface. And Alex went over some of this, but I'm going to go into a little bit more detail on each aspect of the results and how you use the interface. So you just start typing and what comes up will match what you've typed. And I'm going to choose influenza A virus, but this is true for any pathogen or any source organism. So if you're interested in allergens, you can type Timothy grass and it'll show up. If you're interested in bacteria, you can start typing and it'll show up. So any source organism of an epitope, use this search box, and it's a multi-select. So I picked one thing, so you see a 1, but you can pick several things at once, just so you can pick several diseases or several different hosts and perform multiple queries all at the same time. But because you can build your query using any or all of these boxes, you can use the default search and not pick anything, or you can pick something from each box. So when you've completed picking what you want to be in your initial query, you then hit the green search box at the bottom of the page, and that takes you to the results page. And I'm going to walk through quite a bit of detail on what you see on this page. So bear with me, it's a lot of slides, but the slides will be available at the end if you want to look anything else up after the fact. So when you first land, what you get are these current filters up here at the top, and that shows you what you've done so far. So I only picked influenza A virus as the source, and by default, positive assays is checked. If you want to include negative, you can add that, but I just left it at the default. So when you land, the pending filters panel over on the left-hand side, it gives you additional search parameters that were not on the homepage because we just can't fit everything on the homepage, and we do have many other ways you can search, so we want to make them available to you. So to organize those better and make them more efficient, you first land on the default, and in the default, you gain a little bit in each of these search panes. So for epitope, what you gain in the default will be you now can search for data where there's 3D structures. So all epitopes for which the IEDB has a 3D structure, if you click this checkbox. We also add the ability to search by post-translational modifications of linear peptides. And this is a drop-down list of all the ones we've encountered so far. And then in the epitope source box, just like the homepage, we have organism and antigen, but we also now have the ability to search for analogs, mimetopes, and neoepitopes of what search parameters you've picked elsewhere. So if I checked analogs, I would include non-natural analogs of influenza A epitopes in my query results. And then we gained 2 new search boxes. One is receptor and one is reference. These were not on the homepage at all. So the receptor is giving you all data for which we have a TCR or BCR sequence. So if the authors provide this, we always capture it, and we capture as much or as little information as they provide. If they give us CDR1, 2, or 3 sequences, full-length nucleotide or protein sequences, we will capture all of that, and you can search receptor type, chain type, and the regions. If you've sequenced a CDR3, you can enter it here. And again, we also have identity matches, so you can look more loosely and do a blast match to get similar CDR3 sequences and see what they were shown to bind. And then the reference where the data came from, you can search for journal articles or submissions by author, title, date, and PubMed ID. So if you want to look for data from a specific source, you can do that there. And then if I picked the filter option of T-cell, that means you only want to search T-cell specific data. So the receptor search box becomes a TCR search box because now it's only searching TCRs. The assay search box becomes a T-cell assay. search box because it's only searching T-cell assays, and you gain the ability to search direct ex vivo only. So some of our users do not want data where there was an in vitro restimulation step before the experiment. If you want to exclude that data, check this checkbox. And then in the MHC restriction search box, we gain two new things, the resolution and the evidence. And this is how well defined the restriction was by the authors. So some authors may not tell us anything about the restriction of the assay. They may have tested PBMCs, and we don't know if it's class 1 or class 2. Some authors tell us it's class 1. They use CD8 T cells, for example. But some authors know exactly both chains that that epitope is restricted to, and some of our users only want that very precise data. And if that's you, you can choose this, and it'll exclude data where we don't know anything about the restriction. And then the evidence drop down. This is how we know that restriction. So some authors are very clear and precise about how they determine restriction, where others it's more vague. So single allele present, for example, would be a tetramer assay with one MHC and the epitope bounded. So we're certain that that epitope binds an MHC. But MHC prediction, that would be just the authors predicted the restriction and they asserted it, so the curator captured it, but we have no other evidence. So if that's not strict enough for you, you can pick which levels of evidence meet your needs. And then if you do the B cell filter option, you get B cell specific search panels. So the receptor search box is now searching only antibodies, and the assay search box is only searching B cell assays specifically. And you gain one new search feature, the antibody isotype. So if you're only interested in IgE or IgA data, for example, you can choose that here and look at just that data, because we have all sorts of isotypes and we do capture of that when the authors provide it. And the last type of filter option is MHC. And so MHC is either MHC binding assays or MHC ligand elution assays. And you can differentiate that here. And in the MHC restriction search box, just like with T cell, we have the option to limit the resolution. How finely did we know the MHC, and the evidence of how we happen to know what MHC they used or asserted. So back out to the results. So when you're on the results, Alex showed this too. We have these different tabs, the epitopes, antigens, assays, receptors, and that's how you access the details of those experiments that brought you these epitopes. So on the epitope table, there's these headers, and they're all sortable. You can click on these little arrows. And then what a common feature throughout these pages is this details ID, the IEDB ID, and that takes you to the details about this row, about this specific epitope. And so each row has one of those. And when you click on that epitope details, you get a summary, and all of the summaries will be the same regardless of the epitope. The top will have a textual summary giving you an overview of the type of information we have. And then there'll be these detailed assay tables. So if there's MHC ligand assays, they'll show up here, and you can see quickly an overview about the epitope. So for example, this epitope has 78 positive out of 78 tested A0201 experiments, so you can kind of conclude is probably a really good binder for A0201. Where some of these others, and we had a question earlier about negative data, if it's only been tested once and was negative, you may not want to conclude it doesn't bind A0101, but if it was tested 30 times and was negative every time, you definitely would want to make that conclusion. And then this epitope had a very few B cell assays, and it had a lot of T cell assays because it's a well-known T cell epitope. And it definitely works really well to produce interferon gamma in many different contexts. But some of these others, it was only tested a few times, so we don't really know for sure. And then back out to the epitopes page, these funnels, this is where it's really easy and quick to filter your results right on the fly. If you now looked at this and you said, this is the epitope with the most data, it's the only one I want to look at, you could funnel it, and then all of your tabs would be refreshed to only include data for this epitope. But because I did influenza A query, all of these proteins are from influenza A. And if I wanted to limit right here to matrix protein from influenza A, I could do that right here on the fly using this funnel. And then every one of the tabs, there's the export. And Kelly's going to talk later about more details about the export, because we've updated them recently to add more formats and add the ability to sort of customize what columns are included. Kelly will talk about that. And then the next tab over is the antigens tab. And similarly, it has headers that are relevant to antigens. And then the next tab over is where all the meat of the IEDB is. These are the experimental assays, T cell, B cell, MHC. And again, we have the headers with the sortable columns. We have the inline funnels to filter. And then we have these IDs. This is the assay ID for this row. What we're showing you here is just a summary of our up to 400 fields per assay. So we can't pack all that into this table, but if you want to see the details, you click on that ID and it takes you to an assay details page. And these are really long pages and they follow the same format, regardless of assay type or epitope. They always start with the reference where the data came from, and they always have on the right hand side link outs to the other resources. So this one links out to that paper. Then the next section is always about the epitope, and it'll have all the details of where this epitope was in that paper and link outs to the accessions and the taxonomy for that epitope source. And then the first section will always be about in vivo immunization. How did the host come to mount the immune response? It could have been a human with the disease. It could have been a rabbit injected with protein. In this case, it was a mouse injected with the actual epitope, but it doesn't need to be the epitope. It could be a whole virus, and it could be a whole protein. It could be a different epitope that it cross-reacts with. Then the next section is about the assay itself. This is a treated thymidine assay, and this is the link out to the ontology we use for that, the ontology for biomedical investigations. And here you have links to Ubron and cell line ontology for the cell types that were used in this assay. And then there's the MHC information, and here we have links to MHC restriction ontology and what was the assay antigen. So the assay antigen doesn't have to be the epitope. It can be the whole protein if you have epitope-specific antibody binding to a protein. It can be the whole organism if you have a challenge assay showing that immunization with the epitope protected from that challenge. And then for all of the assays in IEDB, we tell you where we got them. This was from figure 2 in that paper. So you can go back and look if you want to see additional details. And then the next tab over is receptors, the TCR and BCR sequences, just like the other tables. And again, we have an ID, and that gives you information about this row and the IEDB groups. Currently, the receptor sequences using CDR3 sequences. So for this row, when you click on it, you get a details about that receptor group. It gives you a textual summary at the top, links to the 3D structures, and then the information that we got from the papers on that receptor. So you can see in 2 cases, the authors sequenced both CDR1, 2, 3 of alpha and beta chains. And then in one of the papers, they didn't provide CDR1, 2, but they did provide CDR3. And so we always will have whatever the authors gave us. And you can see here, this is slightly different resolution. In this one, we have TRBV 1901, and here we have 19. So the curator captures whatever the authors give us, and they may some receptor may bind one epitope, some receptors may cross-react across species. In this case, it looks like these receptor groups are binding that one influenza epitope plus several different analogs of it. So you can see how different mutations do not lose the binding to that receptor. The last tab is references. And this is, where the data came from, and it has the same features as the other tabs with the headers, the funnels, and the link out. So you can go directly to the paper if you want to look at it after looking at what we have. And then I'm going to go through an example and show you the results in that context. So the question was how to see the differences between B and T cell responses for any pathogen. And I started with my query before for influenza. So I was looking at all data related to influenza, and you can see the 2 different tabs, T cell assays and B cell assays. That alone gives you some information about the differences. But you can distinguish between them by using the assay search box and unchecking the type of response you don't want to see. So first I did a T cell search by unchecking B cell MHC. So then the same query for influenza gives me 2,200 epitopes that only have T cell responses. And then the B cell query, I unchecked T cell and added the check for B cell. So I'm only seeing B cell data. I get 1,300 epitopes. And it's entirely possible for the same epitope to have been tested in both B cell and T cell experiment types. And it's also possible to be tested in MHC assays of B cell. We don't police what people publish. We just capture everything. So sometimes there's certain epitopes that have a lot of data in all 3 categories, and sometimes there's epitopes that only have MHC binding data. It's entirely up to what comes out in the literature. And then one of the best ways to see the data, and I think that subsequent talks will show this a little bit more detail also, is on the antigens tab is where you access ImmunomeBrowser, and this really helps you visualize your query results. So you access ImmunomeBrowser using this little graph, bar graph icon. When you click on it, it will plot all of your results along the length of whatever protein you clicked on it next to. So that's why it's on the antigens tab. So I did this for hemagglutinin and compared B cell and T cell. So it will plot whatever your query was. So my query was just for B cell data from influenza A virus. And the blue peaks are positive assays and the red are negative assays. So it can be helpful to see the negative because it tells you if a region isn't blue because it was never tested, or is it not highly positive because it was tested and was found to be negative repeatedly. And these graphs always plot what your query was. So if you wanted to compare human and mouse or two different strains of mice, just redo your query and then you can plot it again and see how they're different. So here we can see how the discontinuous B cell epitopes span a wider range, where the T cell linear epitopes have these very particular peaks. And then I repeated the query again, comparing CD4 and CD8. And again, you can see how the CD4 peaks, the blue positivity, are in specific locations, and the CD8 peak is in a different location. And for different proteins, you get such a wide variety of differences, depending on the biological relevance of that protein and what experiments have been done. And I think this is my last slide.